All right, everyone. Hello, please, can you all hear me?
Yes, sir.
Yes, sir.
All right, so as we do it regularly, will somebody move to share with us what we did last week so that we can take off from there?
Hello, sir. Hello, sir.
Yeah, Godfrey, let's hear you.
Yes, sir. Sir, good morning. Sir, please, last week we did organic biomolecules. Hello, sir. Hello, sir.
Godfrey, I hear
Yes,
you.
sir.
Please go.
Okay, sir. Last week we kindly did organic biomolecules, and we said these organic biomolecules, they are the carbohydrates, the nucleic acid, the lipids, and the polypeptides. And we said that these organic biomolecules, they have characteristic. And we said one of the characteristic is that they are organic in nature. They are organic in nature. And we also learned that these organic biomolecules, they are also highly, they also dissolve in an organic solvent. And we did mention of the benzene and the dimethyl sulfur oxide, the DMSOs. And we also learned something about they exhibit stereoisomerism. And you brought a diagram of fructose and glucose and other organic compounds. And we gave the organic structure of each of the following. And at the end of the day, we said that when we talk of isomerism, is when compounds have the same molecular form, but different structure and shape. That is when we talk of a compound called stereoisomerism. And we also learned that organic compounds, organic biomolecules, they are they also they are highly they react. They highly react in organic solvents. So that was what basically we did last week. And we also learned that we have the glucose and the, yes, sir.
Yeah. I didn't say they react with organic solvents. I said they...
No, they react.
No, they dissolve.
They dissolve in organic solvents.
Dissolution is different from reaction. If you react, you produce new product. And if you dissolve, all right, so please do the correction. But let's hear you.
Okay, and we also said that they react, they highly react to produce less energy, and they produce less energy too, as organic biomolecules. And we learned, and so that was what basically we learned about the characteristic of organic biomolecules. Thank you, sir. Hello, sir.
Hello, Mr. Danso. Thank you
Sir,
very much.
welcome,
Yeah,
sir.
thank you. I wish we could have another person to also help with it. Yes, anybody? If I heard you well,
Hello,
much
sir.
of what you asked was typically an intro to organic biomolecules. Yes, Elijah.
Sir, good morning.
And the Honorable, how are you?
Please, I'm fine, my grace.
Okay, all right.
To add to what my brother said, I think the characteristics, one of them has to do with the organic biomolecule reacting very slowly. So for that matter, there is a need for their reaction to be hastened by introducing an enzyme. So that is to add to that. And the dimethyl solvent that he mentioned too, if I remember vividly, it play a dual purpose. That is basically by dissolving both the organic and the inorganic solvent. So when we look at the glucose and the galactose and the fructose structure, so looking at the structure, that was when you were explaining the stereoisomerism, we were made to understand that they all have the same molecular formula, but when you compare their structures, they have a different structure form. So when we look at and also we talk about the bond. So the bond or the functional group that basically exists between the glucose and the galactose, we say is the aldehyde functional group, which is on the first carbon, and galactose to the same. But when you look at the fructose, the ketones functional group different from that of the galactose and the glucose. So that particular functional group, the ketones functional group, we said that it can be found on the second carbon. Then from there, you briefly take us to the anomers, of which we have the L-glucose and the D-glucose. And you may mention that the L-glucose basically they are not nutritive, so the body cannot metabolize them. But for some weight losing purposes, these days they are adding them to our food. So you take them to really lose weight, but then the body cannot metabolically make use of them. But for that of the D-glucose, they can be metabolized by the body. So for that matter, they don't normally add them to add more carbohydrate to the system. So I think that is what I also grab in addition to what my brother said. Thank you, sir.
All right, thank you, Elijah. Yes, maybe. Can we have a lady contributing to the discussion? Yes, anybody? Joanne. Hello, Joanna.
Good morning, sir.
Yeah, good morning. How are you?
Please, I'm fine, thank you.
All right, nice.
Please, I also want to add up to what Brother
Oh,
Elijah
sure.
said.
Sure.
You also spoke about the L-alanine and then the
Alan.
D, and then you said that the L-alanine is useful by the body. but the D-alanine is not useful by the body. You also make mention of cholic acid, but with that one, you said that it is poisonous. So they join to amino acid, and then to form, is it glycine? Okay, so the glycine joins to cholic acid to form the glyco, is it glycochocolate, or sorry if I'm not pronouncing it well.
Glycocolate.
Glycocolate. Thank you, sir. And then you started talking about the hydrochloric acid, and you started to let us know the importance of it in cosmetic of late. And then you said it is important and for wound healing, and then and it also has anti-aging properties. That is what I also know. Thank you.
You're welcome. All right. So God bless you. Thank you all. So essentially, we started off with organic biomolecules. That's what I gather from your contributions. So this morning, we're going to start with one of the major organic biomolecules in the form of what? Carbohydrates. Are you clear? And thereafter, we'll take the other ones as well as we journey along. So let's start off with, I'm going to share my board with you. But prior to that, let me ask if anybody has a question. Is there anything you have read that you may need clarification for? Is there anything you heard last week that probably you couldn't wrap your minds over? Yes, any question, class? Hello, good people. Okay, so we had said that organic biomolecules have four main characteristics. Number one, they dissolve better in organic solvents. Number two, they exhibit what? Stereoisomerism. In other words, they can exist in multiple structural forms. And then they also react very slowly, but their reactions can be made to hasten by the introduction of what? Enzymes. And then lastly, we had said that they contain carbon, hydrogen, and what? Oxygen. Okay, but they may contain other atoms such as sulfur, phosphorus, nitrogen, and even iron, IUK, or even metals, IUK, like hemoglobin and the rest. But traditionally, all of them have these atoms, carbon, hydrogen, and oxygen in specific ratio. Okay. And we looked at some common examples of organic biomolecules. In fact, the list is very endless. Okay. They constitute a larger proportion of our biomass. Okay. They are widely distributed in our body. So we say they are ubiquitous. They are widely distributed in our cells and by extension in our body. So you find them everywhere, from the crown of your head to the sole of your feet, from the marrow of your bone to the last drop of our blood, from, just name it, everywhere, are you okay, of our being, they occur there. So they are highly ubiquitous within us. And that's why we need to study them to understand them in terms of their nature and their function. Okay? All right. So let's start off with the first one. Number one, I said they are categorized into 4. The first is what? Carbohydrates. The second is what? Proteins. And then the third is what? The lipids. Then the last one is what? The nucleic acid. So let's take the first one. Are you okay? Physiological carbohydrates. Number one, they are organic in nature and contain carbon, hydrogen, and oxygen in the ratio 1:2:1. 1:2:1. In other words, they conform to the general molecular formula CH2O, all in bracket N. Where N takes values from 3. So they are greater than or equal to 3. Okay. So what it means is that physiologically, the simplest or shortest chain of carbohydrates will have how many carbons? 3. So you have 3, then 4, then 5, then 6, IUK, and etc. Number 3, they occur in the body by 2 main ways. In other words, there are 2 main sources of physiological carbohydrates. Number 1, through diet. So we normally call this exogenous source. Through diet. So you can get carbohydrates in your cells through the carbohydrate-rich diet that we take. In Africa, most of our carbohydrate-rich diets contain a lot of starch. So the fufu, the banku, the staples, are you okay? They are so rich in starch, and once we take them, they break down and then they are released into our cells, or they are absorbed and distributed into our cells. Number 2, through metabolism. Through metabolism. Now, through metabolic reactions, our cells are able to produce what? A lot of glucose. The first metabolic process is called gluconeogenesis. So through gluconeogenesis, our cells are able to produce a lot of glucose. which is a common form of what? Carbohydrate. Okay. There's another one called the pentose phosphate pathway, PPP, pentose phosphate pathway. So this process converts normally glucose or hexose sugar into pentose sugar. The main product for this pathway is usually a common pentose called what? Ribose. And you know ribose is used in producing RNA. Okay, so it's very, very important for cellular process. Okay. Number 4. Please, I hope you are following me.
Yes, sir.
All right, so point number 4. Simple forms of carbohydrates are very sweet. Simple forms of carbohydrates are very sweet. However, complex forms are insipid. That is, they are not as sweet as the simple forms. So simple forms of carbohydrate like glucose, like fructose, these are very sweet, but complex forms or polysaccharides like starch, like glycogen, and etc. are not sweet, but rather insipid. Let me ask you this. Is there anybody among you who has ever tasted custard without sugar? Hello, class. Hello, please, can you hear me?
We can hear you, sir.
Yes, sir, we can.
Has anybody ever tasted custard without sugar?
No, please.
You said the anybody cocoa.
Yes, sir.
I'm sure there may be one or two of you who have ever tried this. It's not too scarce. I think it's a common thing. Custard. You know, custard is pure starch. Are you okay?
Yes,
And
please.
yes, I would wish somebody would share with us their experience. How does it taste? Cooked custard without without sugar.
Yeah,
Yes.
me, my own, I always add sugar, so I don't know how the taste is without
Well, let's
sugar.
did you try it before the sugar? That's what I mean.
Mm, it's tasteless though, yeah.
It's tasteless, very good. That's what I wanted to hear. So
Yeah.
normally yes. Normally custard is very tasteless, but custard is starch. And this is carbohydrate. But compared with glucose tonic, how does glucose tonic taste? Hello?
Sweet, sir.
Yes. How about glucose tonic?
The
It's
glucose
very
tonic is
sugary.
very sweet. Even the raw, dry form of it is very, very sweet.
So
And
glucose and
sugary.
yes, glucose and custard are all carbohydrates. I could see that glucose is very sweet compared to what custard.
Yes,
And this
sir.
battery point I'm making, glucose is a simple sugar. And usually for simple sugars, they are really sweet to taste. That's why we call them saccharides. Saccharides. Are you okay? The term saccharide means what? Something that is sweet to taste. Are you okay?
Yes, sir.
And this is one of the reasons why, even currently, they are still employed in pharmaceutical formulations. Are you okay? There's what we call corn syrup. Are you okay? Corn syrup, which is a concentrated form of fructose and glucose together. I don't know the formula anyway, but it's a very, very sweet formulation that is used to sweeten syrups, are you okay, for babies and for other pharmacological formulations, are you okay? Most especially the multivitamins, both for convalescent patients and for children. You know how children easily gravitate to something that's sweet, are you okay? So that's about it. And even this corn syrups, I think sometimes some of them are used even in cake production, apart from the sugar added, are you okay, and other confectionaries, okay, and these and etc. And it's all because of their sweetness, are you okay? So it's very important. But mention should also be made that not everything that tastes sweet contains simple carbohydrates. In our modern world of things, um, we've been able to synthesize substances which equally taste sweet, but they are non-carbohydrates. We call them artificial sweetness. Are you okay? Artificial sweetness. Common example is the aspartame, the oxysulfate K, and saccharin. Are you okay? They are used widely, especially in formulation of fruit juices. You know, natural fruit juice is quite expensive. And so a lot of the imported fruit juices are mere water formulations. So they have water, they have sweetener, then they have color, and then they have the aroma, and the preservative. So one of the things they use in sweetening is the chocolate. And they help to sweeten up the formulation. Marianne, your hand is up. Hello, Mrs. Papu. Hello, Marianne. Hello. Okay, so maybe Marianne is struggling to hear us. So That's point number.
I was taking off. Please, I wanted to find, you said when they are found in diet, it's called an exogenous, is the exogenous source. So I wanted to know what it's called when it's found through metabolism.
That's endogenous.
Oh, yeah, thank you.
Welcome. All right. Yes. Any other question, please, or concern? Okay. So point number 6. Point number 6. Carbohydrates can be grouped or categorized by 3 main criteria. In other words, biologically or physiological carbohydrates can be grouped or categorized using 3 different criteria. Criteria number 1, you can use the number of carbon atoms that are present in the carbohydrate. The number of carbon atoms present in the carbohydrate. Okay. It looks like
my network took me off, so I.
Hello, lady. Hello, class. Please, can you hear me?
Yes, please. We can hear you. We're off, but we can hear you now, sir.
Okay. All right. So what I was saying was that physiological carbohydrate is very diverse. When I say diverse, I mean it comes in different forms. Are you okay? So to be able to understand them, we group them, are you okay, into smaller groups so that you can look at their common feature and then describe them. So that's where the categorization comes in. And I'm saying that we can use 3 different criteria for grouping or categorizing them. Are you okay? The first criteria is to employ what? The number of carbons that are present in the molecule. Are you okay? So there are those ones that share equal number of carbon atoms. They may be different altogether, but they may share equal number of carbon atoms present in them. So number 1 is what? The number of carbon atoms that are present in the molecule. Usually, like I said from the molecular formula, if the number of carbon atoms present in the molecule is, or let's say, like I said earlier, the smallest or shortest chain of carbohydrates, the shortest chain of carbohydrate should have what? A minimum of 3 carbons. Should have a minimum of 3 carbons. Okay. So those ones with 3 carbon atoms, we call them triose sugar. Triose. Trios. Please forgive me, my board is not loading. So I have to wait for a while. Don't know what's wrong. So those with 3 carbon atoms are termed as what? Triose. Okay, triose sugar or triose carbohydrates. So we spell it as TRIOSE. TRI. Triose sugar. Then there are those with 4 carbon atoms. We call them tetrose. Tetrose. We call them tetrose sugar. So if they have 3 carbons, it's triose. If they have 4, it's tetrose. Are you okay? If they have 5, it becomes what? Pentose sugar. So if you hear pentose, like I just spoke about, are you okay? With regards to the pentose phosphate pathway. Okay. I don't want to go. I don't need. Hello, please, can you hear me?
Yes, sir, we can hear you now.
But first, we're not hearing anything.
Okay. I don't know what's wrong with the network. It just went to. So I was saying that the first criteria you can employ in categorizing carbohydrates is to what? Look at the number of carbon atoms that are present. And I told you that the minimum number of carbon atoms you can consider for a carbohydrate is what? 3. Are you okay? 3. And usually when a carbohydrate has 3 carbons, we say it's what? A triose. Are you okay? Triose. If it has 4, it's called what? A tetrose. If it's 5, yes, we call it pentose. Are you okay? If it's 6, we call it what? Can somebody suggest it? It's a hexose. Thank you. Hello, please, can you see my screen?
It's black.
Black.
I'm trying to share a page with you.
You can see it now.
Okay, so like I said, if a carbohydrate has what? 3 carbons, it is called what? A triose. Please, can you see my cursor?
Yes, please.
Hello. Please, can you see
Yes,
my
we can
page?
see.
Yes,
Okay.
please.
So if it has 3 carbons, carbon number 1, number 2, and number 3, then it's a triose. An example is what? Glyceraldehyde. Are you okay? There's another one called dihydroxyacetone. DHA. Dihydroxyacetone. And then you have glyceraldehyde. Okay. And then if it has 4 carbons, it is termed as what? A tetrose. An example is what? D-triose. Triose. Okay. So triose. There's another one called erythrose. Okay. Erythrose. E-R-Y-T-H-R-O-S-E. Erythrose. Okay. So triose, erythrose, even erythrulose. Erythrulose. E-R-Y-T-H-R-U-L-O-S-E. So erythrose, erythrulose, triose. These are all forms of tetrose. Then we have pentose, which has 5 carbons. Are you okay? 1, 2, 3, 4, and 5. And a common example is ribose. Another one is called ribulose. Ribulose. Okay. Then hexoses. These are the very commonest ones, and they abound a lot. An example is glucose. Another example is galactose. Then you have mannose, and then you also have sorbose, and you have fructose. All these are common examples of hexoses. Then we have heptose and etc. Are you okay? All right, so this is the first categorization employing what? The number of carbon atoms present in the molecule. The second criteria is to use what? Is to use the principal functional group present in the molecule. Is to use the principal functional group which is present in the molecule. Now, there are two main principal functional groups. The first is called a ketone. Are you okay? And then the second is called aldehyde. All carbohydrates that contain the ketone functional group are called ketose sugar. Ketose sugar. Those ones that contain the aldehyde functional group, they are called what? Aldose sugar. Aldose sugar. So an example of an aldose sugar is what? Glyceraldehyde.
Good morning.
An example, a ketone sugar.
Yeah, but a drum man, but on one and ten. But on a card, no one ten.
An example of a ketose. An example of a ketose sugar is dihydroxyacetone, DHA. Or what you, another example is fructose. Are you okay? So you can either classify them as aldose or as ketose, depending on which functional group is present. So let me show you an example. If you look at this set, these are all carbohydrates, but look at them carefully. What do you see? You can see that these two have the aldehyde functional group. So they are all called aldose sugar. So glucose is an example. Galactose is also an example. But fructose rather has a ketone. So it's a ketose sugar. If you look at them, you can see that they all have the same number of carbon atoms. That is 6. This one has 1, 2, 3, 4, 5, 6. So we call it as what? An aldohexose. It's a hexose with an aldehyde functional group. There's also an aldohexose, and then there's a ketohexose. Are you okay?
No, sir.
Yes, any question?
Yeah, please come in.
Yes, I said that.
Please kindly go over for us.
I said that if you look at these sets, this is glucose, and this is galactose, and this is fructose. In terms of functional group, you can see that the glucose and the galactose have the same functional group. This one and that one. And these are termed as what? Aldehyde. And I told you that any carbohydrate that have an aldehyde functional group is called an aldose. Aldose. So glucose is an aldose sugar. Galactose is also an aldose sugar. But if you take fructose, it rather contains a ketone. Are you okay? It contains a ketone. And so it's a ketose sugar. Now, in terms of the number of carbon atoms, they all have the same number of carbon atoms, 6. Are you okay? So if you have 6 carbons, then you are a hexose. So we can refer to glucose as what? An aldohexose. Aldo means what? It has the aldehyde functional group. Hexose means it contains 6 carbons. Galactose is equally an aldohexose, but fructose is rather what? A ketohexose. Please, are you okay? Hello, Cecilia.
Sir, please, good morning.
Good morning.
Sir, please, the problem is, how do we identify the group to be a ketone functional group? I mean, the qualities or properties that we can use to identify the group being a part of the fructose functional group.
Okay, thank you.
Welcome.
These are not physical, but rather chemical. Are you okay? So you can use taste or color or how it feels in the hand or any physical sign to be able to distinguish between them. These are purely chemical. Are you okay? That is why I have drawn the structures to point you to the identifiers. So in the structure, if you see such an organization, so if you see these ones, this arrangement is an aldehyde. So you have C double bond O and then at the same time another H. And that's an aldehyde. Okay. If you see C double bond O, but you don't have an H, but rather two different alkyl groups, then that becomes a ketone. Sisi, are you okay? Hello, Cecilia.
Sir, please, I'm okay. Thank you very much.
You're welcome. All right. So let's go. Then the third criteria is to use the number of simple sugar units. Number of simple sugar units. So in this context, there are those ones which exist as just one simple sugar unit, which we call monosaccharides. Are you okay? Monosaccharides. They are just single. Mono means one. So they are single sugar units. Are you okay? And we call them as the simple sugars. Glucose, fructose, galactose. These are all referred to as simple sugars because they exist as simple and single units. Are you okay? Then these single sugar units can join through what we call glycosidic bonds. Through what we call glycosidic bonds. And form what we call disaccharide, oligosaccharide, and what? Polysaccharides. Are you okay? So common examples of disaccharide is what? Formed when you have glucose joining with fructose. Are you okay? When they become disaccharide, they form what we call sucrose. This is what is contained in simple sugar, or what we call table sugar. Table sugar. So the table sugar we use for our beverages, for our cocoa, and for other confectionaries and pastries, it's sucrose. Are you okay? Then if you have galactose joining with glucose, they form what we call lactose. And this is what occurs in breast milk. Are you okay? And other forms of milk that we use. Now watch this and watch this. You can see that between the two, there are linkages, but the form of linkage is different. This is considered as beta linkage, and this is rather an alpha linkage. Are you okay? So the type of glycosidic linkage which occur in the disaccharide differ from one to another. Then we have those we call oligosaccharide. They contain between 3 to about 10 or 12 of the simple sugar units. So if it's 1, it's monosaccharide. If it's 2, it's what? Disaccharide. If it's 3, up to about 10 to 12, we classify it as what? Oligosaccharide. Okay. And common examples of oligosaccharides include what we call raffinose. Raffinose. And then there's another one called stachyose. 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 So you have raffinose, Raffinose. Then we have stachyose. Then we have what we call verbascose. 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 Okay. These are all considered as what? Oligo.
Galactose is glucose is lactose.
Okay.
Sir, please, can you go over again?
Which one?
Sir, please, can you go over the examples again?
I said examples of oligosaccharide include number 1, raffinose. Raffinose. Number 2, stachyose. Number 3, you have what we call verbascose, and etc. Okay. Now, most of these occur in vegetables. Okay. Let me suggest this, that a lot of, very few oligosaccharides occur physiologically. The commonest ones which occur physiologically are those ones that act as blood group antigens. Okay, those ones which act as blood group antigens. So I want to show you the structure of what we typically call the blood group antigens, so that at least you will see how the arrangement occurs. And then we also have what we call the polysaccharides. The polysaccharides have about 20 and more. 20 and more of the simple sugar unit. 20 and more of the simple sugar unit. And common examples include what I'm sharing with you: cellulose, starch, glycogen. In our body, we don't have cellulose. Okay, we don't have cellulose. Cellulose usually occur in plants and in wood. But starch from our diet, and then glycogen is what occurs in our body. So usually excess carbohydrate is stored, as what? Glycogen. There are 2 major organs that store the highest amount of glycogen within our body. Number 1 is the liver, and number 2 is the skeletal muscles. The liver and the skeletal muscles. The liver and the skeletal muscles. These store the highest amount of what? Glycogen in the body. Now I want you to look at the nature of the glycosidic linkage and compare them between cellulose, starch, and glycogen. Now let's start off with starch and glycogen. Look at them. What can you say about the bond? You will realize that if you look at them carefully, they tend to have identical bond structure. Okay. And this is why the enzymes that are able to digest starch are also able to digest what? Glycogen. Okay. So glycogen is not a difficult thing for the body to manage it. Okay, just as it manages starch. But compare starch with cellulose, you could see that the bonding is different. Here you have most of the molecules in what? Beta configuration. Okay. And so a lot of the enzymes that are able to break starch down cannot break cellulose. And this is why vegetables and leaves do not form part of our nutrients, apart from the vitamin source, apart from them acting as what? Vitamin source. They are not a good source of carbohydrate for us, even though they contain a lot of carbohydrates. And the reason is because our body enzymes cannot break these beta linkages. You cannot break them down. And so if you can't break them down, you can't make use of it. Okay, so once you take in the leaves, they go into your gut and they come out as part of fecal matter. Okay. Please, are you okay?
Hello, sir. Please, what again did you say was the link between the galactose and glucose forming the lactose?
I said it was a beta glycosidic linkage.
Okay, sir. So it's the same as that of the cellulose you are talking about.
Yes, but this is different because if you look at lactose, it involves what? Galactose and what? Glucose. But in the case of cellulose, it only involves glucose. But the glucose are all in beta form, not alpha. Hello, Joanne, can you see that?
Yes, sir. Thank you.
So that's what makes galactose different from what? Sorry, it makes lactose different from cellulose. So yes, the bonding may be beta, but the components are different. In the case of galactose, you have what? The glucose is in what? Beta configuration. If you look at this, I'm talking about, this is glucose in alpha configuration. Are you okay? Please concentrate on this OH where my cursor is. If it flips up, then it goes into beta. So this is also beta glucose. So you have alpha and beta. Now the body does better with the alpha glucose. Are you okay?
Yes, sir. So, sir, please, is the position that has changed it from alpha to beta? I mean, moving from down to up.
Yes. Yes, it's a flip.
Okay, thank you.
That is why earlier I said that they exhibit what? Stereoisomerism, if you remember. Have you gotten it?
Yeah, yes, please. Yes, sir.
Yes. And this reaction is called mutual rotation. It's a very spontaneous process. In other words, if I prepare glucose tonic, which is in the alpha form, and leave it standing for a while, within a short while, it can reconvert to the beta form. Are you okay? So that's one of the reasons why a lot of the glucose tonic, if you read on the are you okay? If you read on the can, they indicate that you serve it chilled and you don't keep it in what? Hot condition. Because once you do that, there can be this reaction. This reaction can be facilitated. Are you okay? And then again, in the lab, when we are trying to assess the glucose homeostasis, the effectiveness of glucose homeostasis in an individual who is suspected of having diabetes, that's a test we do. We call it OGTT, oral glucose tolerance test. So usually what happens is that you prepare a glucose solution after an overnight fast, and then you administer it to the patient, and you can monitor their blood glucose level over, I think, about 3 hours. Okay, usually minimum. So first, before you administer, you take the blood glucose. Then after 30 minutes, you take their blood glucose. Then after another 30 minutes, you do it. Then after 1 hour, and then the following hour. And then you plot a graph. Are you okay? Now, to ensure you would realize that normally, as a quality control mechanism or technique, you make sure that the glucose you are giving to the patient is not a leftover, but a fresh one. Because if it's a leftover, chances are that it may have converted, are you okay, from the alpha form, which is more usable by the body, to the beta form, which is less usable. And usually, if it's less usable, it is not absorbed so much by the gut. Are you okay? So you give it to the patient and their blood glucose level is not rising so much. So you may be tempted to think they are normal glycemic. Are you okay? Or euglycemic. But they may probably be a full-blown diabetic. Are you okay? So we advise that you prepare the glucose tonic fresh while the patient, and then you administer it to them. Hello, Joanne, are you okay?
Yes, please. Sir, please,
right.
Sorry, not to draw the class back, but in the case of the glucose thing you are talking about, when they give it to the patient, what are we looking at? That the body will work on the glucose and then we'll see the end product.
Yes.
Or what are we looking
Not
at?
the end product. If somebody is diabetic, the point in question here is that they cannot regulate their blood glucose level very well. So there's a problem with glucose homeostasis. Are you okay? So normally when you give it to them, you would realize that their blood glucose level will rise. For a normal person, you expect the blood glucose level to rise and then fall because of insulin secretion and because of effective response to insulin, which enables the body to absorb most of the glucose from the blood. But for diabetics, are you okay? Because of insulin resistance, sometimes insulin may be produced all right after the glucose level has risen. but the body is not responding to the insulin. So it is not absorbing the glucose from the blood. The glucose remain in the blood. So you see that if you plot the graph, you see that the glucose level will rise, and instead of falling, it remains flat over time. Sometimes it may fall, but not so significantly. And that tells you that the patient may be struggling with glucose management. And so they may either be in the pre-diabetic stage or a full-blown diabetes. Please, are you okay?
Yes, sir. Thank you. Thank you so much.
Welcome. You're welcome. All right. So that's about how carbohydrates are classified. Are you okay? You can classify them in terms of what? The number of carbon atoms. Number two, they can be classified in terms of what? The functional group present, either as ketose or as aldose. And then they can also be classified in terms of the number of simple sugar units that are present, are you okay, in them. And in this wise, they can be categorized as what? You have monosaccharides, which are the simple sugar units, disaccharides, oligosaccharides, and what? Polysaccharides. I said that the monosaccharides are largely glucose, fructose, mannose, galactose, and sorbose, and even ribose, and the rest. These are considered as simple sugars. Then you have what we call the disaccharide, which is formed through glycosidic linkages. So if you look at my diagram here, this is a glucose in the alpha form, if you can see. So there's alpha glucose, and there's also a beta glucose, can you see here? And then they join together. Now, during the formation of the bond, there is a loss of water. So we call this as condensation polymerization. So OH from the alpha carbohydrate joins with an H from the beta. And then once they are removed, they create a space for the linkage. Are you okay? Through oxygen. Are you okay? And if you see here, the bond is between carbon 1 and carbon 4. Carbon 1 in the alpha glucose and carbon 4 in the beta glucose. So we call the bond as what? Alpha 1,4 glycosidic linkage. And this occurs in maltose. So anytime we talk about enzyme digestion, we are talking about an enzyme that will attack this bond. So the enzyme maltase during digestion will attack this bond and break it so that we can have glucose and glucose. That's why we say that the digestion of maltose produces glucose. Are you okay? All right. And the enzyme is capable of attacking what? Alpha 1,4 glycosidic linkage. Okay. All right. Is there any question? All right. Now, let's look at the physiological role of carbohydrates, and then later I'll come back and talk about a few things. So, why are carbohydrates important in our body? They are important for several reasons. Number one, they are an important raw material for the synthesis of other carbohydrates. They constitute what? An important raw material for the synthesis of other carbohydrates. So if you look here, you can see glucose can be used to produce what? Glycogen, which can be stored in the liver and in skeletal muscles. Are you clear? which can be stored in the liver and in skeletal muscles. Now, when glycogen is stored in the liver and in skeletal muscles, they are stored for two different reasons. Are you okay? So when carbohydrates are stored in the skeletal muscles, they are reserved for energy generation, immediate energy generation during fasting. Are you okay? So when an individual is fasting, let me ask you this. Have you realized that sometimes you can go through a whole day from morning till, let's say, evening? Are you okay? Without eating, and still you will feel so strong to be able to do everything you want to do, even without feeling hungry or weak. I don't know if it has ever happened to you. Hello?
No, sir.
Yes,
Have you
sir.
ever? It may not be because you are fasting or you have intended not to eat. Sometimes you just forget about yourself because the day demands or the day appears to be very busy. And you just carry on through the day without eating. And you yourself, you even forget, you don't feel hungry, you don't feel tired, and you go through the day. Sometimes it may even take another person to remind you that you haven't eaten in the day. I don't know if it has ever happened to any of you. So during such time, yes, the energy with which you are doing what you are doing or what you were doing would come from what? A glycogen reserve within the muscles. Are you okay? So what happens that during that fasting moment, when you are not taking glucose directly from diet, what happens that the body switch over, are you okay, to borrow from a reserve of glycogen in your muscles, okay? And then they break the glycogen down, and then the glucose that are generated are burnt within the muscle. They are burnt within they are not released outside, okay? They are burnt within the muscle so that they can generate energy to drive muscular activity. So you still keep moving and doing everything possible. But if glycogen is stored in the liver, what happens is that in the liver, the glycogen is broken down to release a lot of what? The glucose. And this glucose is released into the blood. Are you okay? During fasting. And the essence is to ensure that while you are fasting and haven't taken in glucose, your blood glucose level will not decrease significantly so that you fall into hypoglycemia. So we always say this in physiology that when the glucose store glycogen, it is to maintain euglycemic level during fasting. Euglycemic or normoglycemic level during fasting. The essence is to ensure that when you are fasting, your blood glucose level doesn't fall significantly into the hypoglycemia. Okay, but ensure that constantly there is glucose to replace the ones that are taken in. Mind you, when you are fasting and not eating, your vital organs are not fasting. They need glucose to continue to do what they do. Your brain, your heart, your kidney, your lungs, these continue to work to sustain you. And in as much as they are working, they will need a lot of energy. And to get energy, they need fuel. Are you okay? And the main fuel for the cells of these vital organs is what? Glucose. So they'll be taking a lot of glucose from your blood. Are you okay? And so the blood glucose level is supposed to fall, but it doesn't fall. Why? Because as they take the glucose from the blood, the glycogen reserve in the liver are broken down. to generate glucose, which is fed into the blood to replace the lost ones. Okay, to replace the lost ones, so that constantly your blood glucose level remains steady or flat through the day until you eat again. Are you okay? At another time. So that's basically what happens. Are you okay?
Yes, sir. Sir, please,
All
I
right.
want to ask.
Sure, ask your question.
I want to know the reason behind intermittent fasting and weight loss.
Okay. The point is that usually it is assumed that if you eat regularly all the time, sometimes instead of excess carbohydrate being converted to what? Glycogen. You know, glycogen is not for a long term, okay, but rather for what? Short term. Are you okay? So let me ask you this. If I give you 100 Ghana now, what are you going to do with it? If you realize you don't have any pressing need to spend the money, you will keep it with you because 100 Ghana is not too much. Now, if tomorrow I give you another 100 Ghana, supposing you don't have a pressing need, what do? You keep it. So you have 200 Ghana. If the next day I give you 500 Ghana, which you may not spend, it becomes what? 700. If I give you another 500 Ghana, how much will it be? It will be 1,200. I'm sure you are following me. Okay. Now, if this continues, at a point you see that the amount of money you have is quite substantial. So what do? Instead of you keeping it with you, you take it to the bank. Are you okay? Why are you taking it to the bank? Because you think that you won't need it immediately. So that's what happens. In the immediate term, the body stores energy in the form of what? Glycogen. But in the long term, it rather will store it as what? As fat. So sometimes carbohydrates can be converted into fat and stored. And that is where the problem lies. Because if it's converted into fat and stored, then it means that you are putting on a lot of what? Weight. Are you okay? Through adipogenesis. So what happens with intermittent fasting is to ensure that you clear the backlog. So when you fast maybe for a day, once in a week, you clear the backlog of glycogen reserve so that you don't switch into restoring them in fat. Are you okay? And that's why we encourage that as a strategy for weight management. Are you okay? Hello, lady. Hello, class. Are you fine?
Yes, sir.
Yes, sir.
Right. All right. So that's about carbohydrates serving as raw material for the synthesis of other things. Now, beyond glycogen production, sometimes carbohydrates can be used to synthesize fat, like I just said. Are you okay? And it can also be used. I don't know why my board is not loading, but I wanted it to load so I could share a few things with you. It can also be converted into an intermediate called acetyl-CoA. This compound can be used to synthesize cholesterol. Okay, cholesterol, which can further be used to synthesize steroid hormones, build cell membranes during reproduction, and etc. Okay, so carbohydrates are very important as raw materials. In fact, they are indispensable as raw materials. Okay. Okay. Number two, Number two is that carbohydrates can also be used, or they constitute what we call structural components in our cells. Carbohydrates constitute what? Structural components of what? Of our cells. Hello, please, do you have my slide?
Yes, sir.
Yeah.
Okay. They constitute important component of what? Our body structure. Okay. How do they contribute to that? They serve as integral components of our cell membrane. And you know, our body is made up of cells. So whatever is a component of the cell becomes a component of our body structure. So if you look at them, this is a typical plasma membrane for most of our cells. Are you okay? This is what we call the phospholipid bilayer. And you can see projecting from the surface of the cell. What do you see? You have a lot of what? Complex carbohydrates, glycolipid, and glycoproteins. These serve three important purposes. Number one, they act as receptors. Let's say together, receptors.
Receptors.
Yes, yes. Number two,
Receptors.
okay, thank you. Number two, they also, number two is that, yes, they also act as well as transporters. They help the cell to pick up molecules and then transport them into the cell or outside of the cell. Are you okay? So number one, as receptors. Number two, as transporters. And number three is that they also help the cell and confer on the cell unique identification. They help as an identifier of the cell. Different organs in our body have different identifier molecules on their surface. That is why we can separate the red blood cell from the white blood cell, even though they are all in the blood. Are you okay? Because on the surface of the red blood cell, we have a unique identifier molecule. So you can develop antibodies which will target that and then pull it out of the blood. Are you okay? So you do the separation. We call it flow cytometry in molecular medicine or biomedicine. Are you okay? So you use that technique so that you can separate the cells individually. That is why sometimes when a particular organ is transplanted, the body can fight it because the body knows the unique identifier of self molecules. Are you okay? So the one coming in, if it has a different identifier, they can know that this is not part of us, and so they reject it. Okay, so this is very, very crucial. Very, very crucial. If you look here, you can see that the cell receptors are being exploited by what? Microorganisms to be able to invade our cells. Okay, to be able to invade our cells. So this is one of the techniques that is employed by microorganisms to be able to what? Invade or enter into various cells and cause what? Infection. Okay, so let me give you an example. The HIV virus is in the blood, but it doesn't affect the red blood cell, which is also in the blood. but rather affect the white blood cell. Why? Because it is able to bind to receptors on the white blood cell and then enter into the white blood cell to cause what? Damage. But it doesn't have a reasonable receptor on the surface of the red blood cell. So it is with the red blood cell, but I can't infect it. Are you okay? Hepatitis virus occur in the blood. That is why if they want to diagnose hepatitis, what do? They use your blood, but it doesn't affect any of the blood cells. It doesn't affect your red blood cell. It doesn't destroy your white blood cell. Are you okay? But it affects your liver. Why? Because on the liver, there is a particular receptor that this enzyme, this microorganism can bind to, by which it can invade into the cell. Are you okay? I am going to play you a short video on how microorganisms exploit what? This phenomenon. Are you okay? To be able to invade into cells. And then maybe after that, we'll bring the class to a close. So, thirdly, apart from carbohydrates serving as raw materials for the synthesis of other biomolecules, they are also important in what? In the structure of our body. Are you okay? The third thing is that circulating carbohydrates within the blood can be used as biomedical marker. Are you okay? They can be used as what? Biomarkers for various what? For diagnosing various diseases. A common example is what? Is diabetes. If you suspect anybody to have diabetes, what do? You check their blood glucose level. Okay. You check their blood glucose what? Level. So what it means that circulating carbohydrates can be used to diagnose diseases. Are you okay? The fourth thing is that they also help in determination of our blood group. I told you that the blood group is determined by an oligosaccharide chain. Are you okay? Within our body. And this is purely genotypic. So you can't change much of it. Are you okay? And a particular arrangement of oligosaccharide will give blood group A, whereas a different arrangement will give rise to blood group B, AB, and what? O. Are you okay? So all that we talk about as blood group antigen is not anything strange. It is just an arrangement of what? Oligosaccharides on the surface of the red blood cell, which confers that unique blood group to the individual. Okay. I think my system is frozen, so the video is not opening. But I wish it would open very soon, so you can. And then the fifth one is that carbohydrates, some carbohydrates have been implicated in what? Aging. Are you okay? In aging. Example is what? The D-galactose. Excessive amount of D-galactose buildup have been implicated in causing organ degeneration. Organ degeneration. So they cause the nerves to degenerate, giving rise to neurodegenerative disease like Alzheimer, Parkinson's, and the rest. Are you okay? All right. And then they can also cause.
The fifth one again.
Hello. Yes.
Please, the fifth one again.
The what?
The importance of
Hello, lady.
carbohydrates, the fifth one. Please, the
I
importance of the
said,
carbohydrate. I said the fifth one.
yes, I said that they have been implicated in what? Or they have been implicated in organ degeneration and aging. An example is what? D-galactose. It's been suggested from research that excessive buildup of D-galactose in the blood can trigger organ degeneration. In fact, studies have implicated this compound, which is a carbohydrate, in what? Degeneration of the heart and degeneration of the nerves, which are all features of what? Aging. Are you okay? So that's that. Lady, are you okay?
Yes,
All right, so
please.
let me share this short video with you, and then let's see what you make out of it. Stop that. Okay, please, can you all see my screen?
Yes, sir.
All right.
2019 novel coronavirus is a newly discovered strain from the coronavirus family that causes respiratory disease. Humans have long been infected by coronavirus, as it is one of those responsible for the common cold. It is a contagious viral infection spread through inhalation or ingestion of viral droplets. As a result, coughing and sneezing and touching
What you are seeing here is the virus.
are primary sources of infection. The structure of the virus has multiple parts. Inside the virus lies a genetic encoding that allows the virus to hijack human cells and turn them into virus factories.
Through what you call mitosis.
The genetic material known as the viral envelope.
We'll look at this in great detail during living cells. But if you look here, what can you see? You can see that the viral structure is made up of what you call envelope proteins. And one of the envelope protein is what? It's a glycoprotein. Can you see that? Glycoprotein is what? It's a protein that is bound to what? A carbohydrate. And this is why I was saying that carbohydrates are important as structural components of various organisms, including viruses. Are you okay? Now let's fast forward. When this virus
proteins. The structure of the 2019 novel coronavirus is a mutation. For more information on this, see the link below. The large S glycoproteins are used by the virus.
Now, this is the virus after you have inhaled it. And this is the cell of our body. Can you see these projections? These projections on the surface of the cell is what we call receptors, the one I showed you. And many of them are glycoproteins or glycolipids. Are you okay? And they project outside so that they can pick up stimulus to be able to communicate to the cell as to what's happening in the outside environment. So let's see how
cells. They most likely attach to angiotensin converting enzymes.
Now, what receptor is this? This is a normal physiological receptor, angiotensin converting what? Enzyme receptor. So this is on the surface of the cell, but it is exploited by the virus. So it's not a viral receptor. It's not meant for the virus. It is meant for angiotensin converting enzyme 2. Are you okay? But the virus will identify with it, bind to the receptor, and then fool the cell to think it's angiotensin converting enzyme receptor. And
the exact mechanism for this is not known. Most likely, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic
damage. So the virus has now entered into the cell and it will cause damage. So inside the cell, this is what we call the nucleus. These are the nuclear pore. This is what we call endoplasmic reticulum. You have mitochondria and etc. So this is the cytoplasm. Are you okay? The virus will now work on the cell and eventually destroy the cell. So this is just to let you know, if I spoke about carbohydrates as well, structural components of our cell and our body, that's what I mean. Usually they occur either as glycoproteins, glycolipids, and etc. And they are all over our body, from the cells of the liver to the kidney to the brain to the nerves to the lungs, everywhere. Are you okay? All right. I want to end my lecture here. So far, we are done with carbohydrates. I want you to write this as a reading assignment, and then I will finally close. But in the meantime, let me see if anybody has a question or clarification you would want me to make. So write this down. Write this down. Read on the physiological roles of the following derived carbohydrates. The physiological roles of the following derived carbohydrates. Number 1, glucuronic acid. Glucuronic acid. Glucuronic acid. Number 2, sialic acid. Sialic acid. S-I-A-L-I-C. Sialic acid. It's a carbohydrate, but it's a derivatized carbohydrate. Number 3, N-acetylglucosamine. N-acetyl, so capital N dash acetyl, A-C-E-T-Y-L, N-acetylglucosamine. N-acetylglucosamine. And then number 4, hyaluronic acid. Hyaluronic acid. Hyaluronic acid. Are you there? Hyaluronic acid. Hyaluronic. Hyaluronic acid. Okay. All right. So thank you very much for coming for today's lecture. I wish all of